So up next, we're pleased to present Dr. James Fink. I introduced him at our commission member meeting 2 days ago, and I was able to say he's one of the youth we corrupted into Dup15q. So James was originally at the University of Connecticut with Dr. Eric Levine, who's actually talking later, and Dr. Storby Chamberlain, and he was one of our Dup15q Alliance funded fellows. And now he is at Q State, and he's going to share some of the great work he's doing in the Dup15q area. Oh no, he'll switch it over.
I thought there were stairs up there. Well, as the talk's getting up on the screen, I just want to thank Dup15q Alliance and the organizers and Vanessa for giving me the opportunity to speak today on behalf of Q State Biosciences. I'm really excited to share the work we've been doing in the context of Dup15q. Okay. So today I'm going to be talking about Q State Biosciences' approach for antisense oligonucleotide-based therapies for Dup15q syndrome. This is a really exciting moment for me personally. As Vanessa mentioned, I did my PhD work in the lab of Dr. Eric Levine. That work was funded by the Dup15q Alliance as a part of a fellowship. I'll talk about some of that work today. And of course, I think Eric will expand on that after this talk. I'm really excited to see those results. But to be standing here talking about an actual therapy and developing a therapy for Dup15q syndrome after coming from some very basic research and looking at cellular models of Dup15q, that's something I would not have imagined 6 or 7 years ago. So at Q State Biosciences, what do we do? When you think about going from a disorder or a disease of interest or a target of interest to developing a therapeutic, we're harnessing 3 core technologies to sort of bridge that gap. The first one is something, again, you'll hear about from Eric Levine, and Stormy talked about this yesterday, and that's using induced pluripotent stem cells. These are cells that come from humans. You can get them from patients. That's the really great power about them. And at Q State, we scale those in ways where we can produce billions of neurons. They're very reproducible, and phenotype them with a really powerful technology in the center, which is something called Optopatch, which is an all-optical electrophysiology platform. So what does that mean? I will get to that in a second, but it's just one way of measuring a cell's function by watching them flash on the screen, changing fluorescence. But I'll sort of cover that more specifically soon. And then part 3 is using AI and machine learning to understand hundreds of parameters associated with how a neuron or brain cell functions. Looking at as one person's eyes, especially in graduate school, I can really appreciate that when you're looking at the function of a neuron, scouring 500 parameters or so is really difficult to pull out very subtle things from millions of cells. And then using that platform to then assess therapeutics. And today I'll talk about actually the development of a therapeutic and how we're going to be using Optopatch and this platform to assess sort of a variety of those therapies. So what's an antisense oligonucleotide? So in short, they're called ASOs, and they are short synthetic sequences that bind to the target of interest by complementary base pairing. So I'm sure from your biology days or even from your talks here, you've heard about the A's, C's, T's, and G's associated with genes, and those bases pair with specific other bases, and that gives them their specificity. And so these are just short versions of those. And Stormy talked about this yesterday. Cells in vivo eat them up, so they can get into neurons very easily. And there's actually, depending on the type of ASO you use, they can do a few different things. One is they can modulate splicing. That's something some companies are approaching. They can also do target knockdown. And then there's ways of also boosting targets through some interesting biology pathways. And as people here associated with Dup15q, you know that the genes on the chromosome 15q region are all increased or doubled or tripled. And so that means there's more of them. And so in this case, we're going to be using a knockdown approach to lower levels of a gene. We don't want to boost it more, right? Because they're already increased. And so we're going to be using an ASO, or I'll be talking about an ASO known as a gapmer. And those have partial DNA chemistry, and they can engage this endogenous pathway in cells that allows an enzyme to come along and sort of degrade your target. And that way it's not around anymore, it can't make protein, and then you can reduce the levels of the thing you're interested in through this mechanism. So it's one of a few different ways you can use antisense oligonucleotides. And again, because of the overexpression of genes in Dup15q, we're focusing on this one particular way of modulating expression. Now, a little bit more background on antisense oligonucleotides before I get further into the specifics of our work. So right now, there is an improved therapy, an ASO called Spinraza for SMA. And these things are delivered intrathecally through the lumbar injection into the cerebrospinal fluid for CNS disorders. So the reason for this is, especially for disorders of the brain, you want to get that molecule into brain tissue. And one of the reasons of using induced pluripotent stem cells for modeling disease is because getting brain tissue is really difficult. And the same is true when you're getting things to the brain, not just taking things from the brain. So this is the route of administration. And interestingly enough, there is already, I think Stormy also talked about this yesterday, UBE3A being explored in the context of ASOs for Angelman syndrome. And that's through, I think, ultragenics and this sort of mechanism on the bottom right there. So I don't spend too much time here because this is the stuff everyone in this room knows about. But the idea here again is that there's many genes duplicated or even tripled in Dup15q. And again, we're targeting, in this case, UBE3A for an ASO approach as a therapeutic for Dup15q. Now, maybe in the Q&A session, we can get to why this particular target, but I think there was really nice, fruitful discussion yesterday during Stormy and Matt Judson's talks about how UBE3A is a really important role player, and although they may not be sufficient in some mouse models to cause behavioral phenotypes in those animal models, there is really strong evidence for it being necessary. And Stormy showed some really nice data of using her isogenic lines to correct specifically UBE3A. And I think Eric's going to have some data exploring ASOs and correction and modulation of UBE3A as well. So anyway, its overlap with disorders like Angelman syndrome, the fact that it's a maternally imprinted gene, it's the maternal allele that's duplicated or triplicated in Dup15q, makes us think that it's going to be a really important target for providing some type of therapeutic benefit to the Dup15q patient population. Now, so I've talked about this technology for measuring how cells function and the cell models and therapies, but when it comes to antisense oligonucleotides themselves, the actual therapeutic modality, what makes Q-State unique? And so at Q-State, we actually design our own ASOs through a design pipeline sort of highlighted on the right that takes into account sequence motifs and specificity and off-target effects. And then we synthesize those ASOs and then use our platform to screen them for both efficacy and then possible off-target effects on cells. So one way to see that is in this middle panel. So here, there are two different sets of ASOs. Those for a sodium channel called NAV 1.7 and those for a sodium channel called NAV 1.8. This particular graph is plotting a function of a neuron. In this case, it's the way it fires under high intensity stimulation. You see the ASOs on the right in red are changing that firing in neurons. They go down, but the ASOs targeting this sodium channel do not change cell behavior. And so this shows that we can specifically modulate a target that's implicated in this particular neuronal function, whereas other ASOs that target something else or related sodium channel do not modulate this particular behavior. So we can measure these really subtle things using our Optopatch platform. So not just screening molecular biology markers, but also the real function of neurons. The other really cool thing that the platform can do. So on the right-hand side are a handful of ASOs that were screened in vivo. And you can see that a few of them cause some issues when delivered in vivo in terms of weakness and spasticity and things like that. Now, if you look at how those particular ASOs affect a neuron's function, so this is called an action potential, you can see that the two that cause problems in vivo are also the same two that cause problems with the action potential waveform. So the action potential is how neurons communicate with each other. So you can imagine if the shape changes, that could be pretty important for the actual tissue. Interestingly, those same two ASOs, when you look at the number of cells who fire action potentials, they're greatly reduced. So there's things that are predictive about the platform in terms of, sort of maybe screening ahead of time, weeding out ASOs that might have tolerability effects in inexpensive in vivo models. So these are just two things that the Optopatch platform can do in terms of applying the therapeutic to then assessing it in vitro in a dish. So now let's talk about the UBE3A ASOs. So the way we're approaching this is screening really large amounts of antisense oligonucleotides against the UBE3A transcript with, again, the goal of, through this gapmer mechanism, targeting UBE3A and getting it to be degraded so that there's less of it. And so on this summary slide is 80 ASOs. It's a subset of the ones we've screened. And every row is a different ASO. And every column is a different experiment in which we tested the knockdown or the reduction of UBE3A. So because there's a lot of UBE3A in Dup15q patient cells, you want to show that you get really good knockdown. It shows that the ASO is effective in this case. And we actually, in this case, screen not just control, these are fibroblasts. We also screen fibroblasts from a Dup15q individual. So we're not just modulating normal UBE3A, but also really high UBE3A. And green on this scale is good. It means that you can get really, really strong reduction of UBE3A. And you can see that just from looking at this heat map. So green's good, red's bad. The colors in between are, those are the ones that are okay at functioning. You can see there's many good candidates here in terms of reducing UBE3A levels. And there's ones that immediately we can knock off the list because they're not as good at knocking down UBE3A, or maybe you need really high doses, and that could actually be a problem when you want to sort of bring these to in vivo models. So this is just a sense of the sort of scale that we're screening and testing different molecule candidates for modulating UBE3A. The other important thing is you want to be able to modulate your target in dose response. So here's, again, a subset of our ASO candidates looking at the UBE3A transcript. So this is not yet the protein. This is the message that the cell reads before it makes protein. So eventually we want to get the protein. I'll show you that, but this is the step right before. And so on the very left-hand side for each cluster of colors, on the very left-hand side per cluster is the sort of highest dose, and on the very right-hand side is the lowest dose. That's represented by these sort of increasing or decreasing black triangles here. And what you want to see is that your highest dose, your UBE3A, which is normalized here at 1, this is sort of cells only, and then these are cells that are applied with the vehicle that we use to deliver ASO. that the highest dose greatly knocks down UBE3A, and that you can get really nice titration of UBE3A levels, at least at the transcript, by decreasing amount of your UBE3A ASO levels. And this is important, right? Because we don't want to bring down UBE3A to 0. People who have no UBE3A have Angelman syndrome. And so we want to be able to really finely tune or show that your ASO is not at every dose going to give you 100% knockdown, because that's something actually that's undesirable in this case. So again, we look in this case for maybe something at the sort of left-hand panel on the very bottom right, an ASO that in dose response wasn't as good as some of the others. So this is again another way to start to select ones that we really like. Now, UBE3A's role is sort of really important at the protein level, which is the sort of 3D form where it goes and targets things in the cells and has a really important role at that point. So we want to show we can knock down UBE3A protein with this ASO. So this again is just a very small subsample. We've done this for all the ASOs that I've shown you so far, but here's some data. So in the left-hand top left and top right are a total of 6 candidate ASOs, where we're just testing, in this case, 2 doses, 20 nanomolar and 200 nanomolar. 200 nanomolar is the same as the screening dose I showed you from the heat map. And what you want to see is that at both of those, you have a reduction in that black mark on the screen, which is a way to quantify protein, sort of aggregating the protein. And that's your UBE3A. We use an antibody that's specific to UBE3A. And then you accumulate it. And then you want to see that in the cases where ASO was on the cells, that gets reduced. So one way you can see that is, if this is your cells in vehicle, so they have UBE3A, when those same cells have been treated with your ASO, there's no mark in that lane, meaning the protein has gone away. So it's been degraded in the way that we've desired. And then we've quantified that on the bottom here. So of course, when you use the higher dose, you get really good protein knockdown. But even for some of these ASOs, you'll see that there's still really nice knockdown at lower doses like 20 nanomolar. So again, another way to potentially rank ASOs based on how well you can modulate UBE3A protein levels. And then in a similar way as the transcript in the previous slide, we've quantified in the heat map the knockdown of the protein at 2 different doses in a heat map. So you can see, there's some candidates on the right-hand side that are quite good at knocking down UBE3A protein. So this means that we've pulled out antisense oligonucleotides that are specific to UBE3A that can not just modulate UBE3A transcript, the instruction before the protein, but we can do that in dose response. So increasing doses knocks it down more, smaller doses knocks it down less. And then we can also do the same type of change, high or low doses, and knock down protein. Now, this is where I think the platform for Q-State Biosciences really comes into play and sort of brings this all home in terms of why is this meaningful, what differentiates Q-State from other players here. And so now I want to bring back to the work that was funded by the Dup15q Alliance that sort of still following me today, really lucky to me. And so this is data from my paper published with Eric Levine. It's on bioRxives. Sadly, we've submitted multiple times and we've dealt with some cranky reviewers, but we have resubmitted, so hopefully it's published really soon. But one of the more striking things that we found, if you just look at these are again action potentials. So these are those spikes I talked about that neurons fire. The controls are in blue, the Dup15q are in purple, and then we use the paternal Dup actually as a control, just in case those other genes that were talked about all yesterday were important for this. And we also looked at Angelman syndrome. We published a paper on that as well. And you can see just from the traces that the Dup15q neurons have way more action potentials than the controls do. And that's just an example of one cell from one control and one cell from a Dup15q patient. But then we took a bunch of lines, different individuals who donated their fibroblasts, and we differentiated them into stem cells and made those into neurons, and quantified all of those here on the bottom right. You can see all the controls behave relatively the same in terms of their level of action potential firing. And when we look at the levels of firing in the 4 Dup15q patients, this includes one interstitial triplication. So they all have really increased levels of UBE3A. You can say, again, there's some variability there, but for the most part, all of them are firing sometimes 2 or 3 times more than the controls. And the Angelman nor the paternal Dup behave in that way. So there's something really striking here at the single cell level And at the time, we had some technology for using calcium as an assay for firing. So you can, similar to what I'll show you for Q-state, we can look at many cells at once, but rather than looking at individual spikes, you can look at an accumulation of these spikes that yield calcium into the cell. And there's two examples, again, from two different control subjects here. So each dot is a time that one cell fired, and each row is a different cell. And you can see there's just a few dots here in the two different control experiments. But when you look at the induced pluripotent stem cell derived neurons from two Dup15q patients, every single cell in the field of view, or at least the ones the sort we can measure, are firing, and they're all doing it together. And this is also really striking. And it's really hard to tie behavior in one, a set of cells that are in a dish, sort of being measured by an instrument, and compare those to what's happening in the real brain of a patient. You can imagine that sort of seizure activity that's going on in the real brain. It could look like this in a dish. So it's sort of interesting to think about this being relevant to actual symptoms or sort of brain functions that are happening in people. And maybe we can use animal models to tie those two things together. And Ben yesterday mentioned a great model for being able to look at this. And there are other Dup15q mouse models or UBE3A overexpressing models. And certainly the stem cells are another way to probe this. So we are licensing these cells in our lab using the Optopatch technology to sort of look at these exact phenotypes with the goal of showing that UBE3A knockdown, which again is really increased in these cells, is going to be able to correct these cellular deficits. And sort of, again, maybe that's another layer in which we can differentiate some ASOs, either in terms of how well they get into neurons or how well they modulate this type of phenotype. So the point of exploring this, when the Alliance was kind enough to fund this work, was to use it for something, and that was to screen therapeutics. So here's a way, 6 or 7 years later, that's sort of coming full circle. Now, this took 7 years to do in graduate school alone. And one of the reasons I went to Q-State was because when you go patch one cell, so doing this in grad school, you take a wire, and you go one neuron at a time, and you patch them. And when you use stem cell derived neurons, they're very variable. So you have to patch lots of cell lines and lots of cells. That's why it takes so long. But because we have this powerful high throughput technology of measuring neuronal function, as I outlined before, we can record up to 300 to 500 neurons in one single instance across 15 seconds, and then up to 500,000 neurons in a day. I did something like 7,000 in 6 or 7 years. So just in 1 hour, we can get the data that took me many years to collect. And again, because these cell lines can be variable, at Q-State, before I sort of switch back from this pretty movie here on the bottom, I'll talk about very specifically in a second. We're taking two approaches to understanding the cellular parts of these and scaling neurons in a way that, again, we can get really reproducible phenotypes. And that's two. One is using cells that actually come from patients, as I mentioned, sort of the cell lines that Stormy has been using and characterizing with Eric Levine, but also CRISPR editing to either induce mutations or correct them. And we do these in parallel just to build confidence in the types of phenotypes we're observing. We want to make sure that what we're seeing is not just some fluke of the particular cell line we're measuring, but really has to do with the true biology. So we want to make sure that we're phenotyping as many cell lines as we can and being as confident in the mechanism that's happening as we can. And again, we do this in a way that the cells we use when we phenotype are exactly the same sets of cells that we use when we screen all of our therapeutics. We don't have to switch batches of neurons in between, which many labs and groups have to do. And so if you want to know what that movie is, each little spark you see on the screen is a separate neuron. And every time you see a spark is when that neuron fires. Now in calcium imaging, you can see this as well, but usually that spark is accumulation of many active potentials. Here, every spark is a single active potential that has the same resolution as patch clamp. And when you do that, it means you can measure many parameters associated with how a neuron behaves. Its height of the active potential, its width, its depth of its after hyperpolarization, which is another important feature, how those active potentials adapt over time when you inject current. You can inject lots of different types of stimuli to check strong stimulation versus weak stimulation versus prolonged stimulation versus short stimulation. There's many permutations of this that can be checked, and all of them have a parameter associated with it. So we can look at this entire space and trying to find differences at the cellular level, and then find the ASOs that don't just correct one or two, but the entire fingerprint. And what do I mean by fingerprint? So for, again, each of these stimulations, we have different parameters we can measure. So here's one example where we phenotype both patient-derived lines and the CRISPR-edited lines. The patient-derived lines on the very left, the isogenic lines were the CRISPR-edited lines. We knocked out the target on the very left. This is for severe monogenic epilepsy. The ones that have a knockout of this gene are in orange, and the controls that are isogenic are in blue. And you see there's this nice white space between those cell lines. That's what you want to see. That means there's a window there that you can correct. There's something different that you can try and screen for. And again, maybe that's just the two cell lines versus the controls. We want to make sure that's not the case. So you can also look at cells that come from the actual patients versus family controls. And in this case, again, the ones that are in orange come from the patients. So again, they don't have this particular target, just like the CRISPR edited lines. And the controls are people who have normal expression of this target. And we see the same window. Now, this one cell line here is a reason why doing this with many cell lines is important, because there is some noise just within the same group, right? So you can't just look at one and one, you have to look at a bunch. So this is really nice and exciting to see this both with the CRISPR edited lines and patient control lines. For this particular epilepsy, there is an approved drug. And so on the right-hand side, the way we sort of view these fingerprints, and this is, there's I think another one on the next slide, but this is a more simple one. You can see that you plot the controls at the center of this, what's called a radar plot. And then these parameters out here are on each spoke, the particular parameter that's different between the controls and the cells who have mutations. And you'll see there's one parameter where there's a really big difference between the orange and the blue at the center. And there's other ones where there's not a huge difference, but there's some tiny white space there. We applied the approved drug for this disorder on the mutant cells and saw, is this related to the disorder? Because if it is, it should be rescued by the drug. And in fact, if you look at the one that has the biggest difference, you see that that went from here, now is much closer to the controls. And so even though they don't have this target, that gene is missing, they're still now behaving more like the wild type cells. And then for the parameters where the difference wasn't so big, you cannot tell the difference between the controls and the drug treated cells who do not have the target. So this is one powerful way to assess therapeutics. And so we can apply this type of technology to testing ASOs, and again, on cellular models of Dup15q. So the whole pipeline here to outline is we have these cellular models that we can use on Optopatch to record these flashing movies that extract these firing of these neurons. It's a really important function of the cells. And then this is a different phenotype. So again, each spoke is a different parameter. This top phenotype is from an isogenic cell line. So the red sort of odd shape is from the mutants, and the ones in the center are the wild types. And interestingly enough, those same exact sets of parameters show the same exact fingerprint when you look at three different patient-controlled pairs in the bottom. So this is a family member and an affected individual, a different family member and a different infected individual, and so on and so forth. So three patient-controlled pairs match identically to the isogenic. So again, building confidence there. And then that gives you something, again, you can correct. And so each dot on this graph, the top cloud, the dark colors are the controls without a drug. The sort of dark blue here are mutant cells without a drug. And then the orange dots here are an approved drug that rescues the disease from here to here. So again, it's bringing them towards the normal levels. And then the light blue dots are screening thousands of molecules to see which interesting new drugs bring them towards this cloud of wells that are part of the approved drug. And so you want to find things that rise to that level. So again, this type of screen can be done really quickly at a level of individual neuron function because of the technology that Q-State Biosciences harnesses. Now, the other critical function of neurons is synaptic activity. neurons connect to one another in the brain, they release neurotransmitter, that neurotransmitter bonds to receptors, and that's actually really, really important. In fact, it's something pretty central, at least a lot of the field believes, to autism related disorders. Many of the autism associated genes, things like SHANK3, even UBE3A in many papers, FMRP, they all have these synaptic phenotypes. So assessing synaptic function is also critically important for assessing therapeutics. And at QState, we've developed an assay for this type of neuronal function that's a little bit different than the last one. So in this case, we use these two different tools in two different cell populations. So that one population responds to, you can imagine half of the room here having earplugs, so that when I give a command, only half the room can hear me, and those people respond, and the other half can't, right? So it's sort of something similar where you express one tool in one set of cells, they can be stimulated, they'll fire an active potential that will trigger this release of this chemical. So again, critically important process for neurons, but we'll read out that response in the other population of neurons with no contamination from the signal that's evoked by the flashing of the light. So it's a really unique assay for assessing synaptic function. And to validate this, we added synaptic blockers. So these are blockers for 2 glutamate receptors. We added them in dose response. You can see the signal without blockers up here gets reduced in nice dose response as you add increasing amounts of, in this case, an AMPA blocker, which affects this very fast component of the synaptic signal. And then more specifically, when you block this other receptor called NMDA, which often affects this later component of the PSP, you see a dose response in this tail component of the synaptic transmission. I presented many phenotypes associated with 3 neurodevelopmental disorders at the commission talk. So we have been using this assay to establish, again, differences between mutants and targets of interest and wild types as a validation for this target. Because of course, if we want to use this for something productive, it has to be measuring what we say it's measuring. So a summary of our findings and next steps: we're advancing an ASO-based therapeutic for Dup15q, specifically for UBE3A. We have identified potent ASO candidates that knock down UBE3A at both the transcript and protein levels, and we can do this in dose response. We want to assess the functional rescue of individual neurons, again, as we have this really strong, unique platform for assessing neuronal function, not just in terms of rescuing phenotypes, but again, assessing some of these off-target effects or perturbation effects that we showed you on that previous slide with the ASOs. And we're pursuing functional rescue in human cellular models of Dup15q. Again, shout out to Stormy and the work that's being done by Eric's lab. And also, we want to explore this in the context of in vivo mouse models. Again, you heard yesterday from Ben Philpott the power of the UBE3A overexpression mouse and some of the phenotypes that the Philpott lab has been pursuing. And so again, our goal here is to advance a transformative therapeutic ASO for Dup15q patients specifically. And then lastly, this is maybe the most brilliant, hardworking team I've ever worked with. There's a lot of people that sort of the thoughts and actual technical execution went into the experiments I showed. This is just not 1 or 2 people. This is really a team effort. So I just want to highlight people on the team that have been working on this. It includes Kathy Harikrishnan, Aishwarya Dandapani, Vehab Joshi, Darit Doshi, Luis Williams, Owen McManus, David Gerber, and Graham Dempsey. So with that, thank you for your attention and time, and I'm happy to answer any questions.
Just a couple of questions. So was there high concordance of UBE3A knockdown in the control fibroblasts versus the Dup15q fibroblasts when you were going through your sort of screening stage?
Yeah, so it turns out in many cell types, we can get the same concordance of knockdown. So yes, control versus Dup15q fibroblasts, same in neuronal subtypes and things like that.
Thank you.
Yeah.
And 2 other quickies. Do you believe that a mouse efficacy model is on the critical path for FDA approval of an ASO targeting UBE3A?
That's a good question. I mean, maybe in the absence of human models, it would be absolutely critical. Maybe if the human cellular models and those functional phenotypes, especially across many patient lines, can be reduced with the UBE3A ASOs, I don't know how critical it would be, but that's something we're gonna have to, get to when we get there. But I guess it depends how that part of so that's why we're doing both. But yeah, that's a great question.
And then in terms of IND enabling tox, do you think that's a multi-species, including NHPs, or is that also, in your view, part of the critical path?
So that would be, so I'm not involved in that level of the sort of project at this point. So it'd be hard for me to say.
Thank you.
Yep.
I have two questions. So if you were to do a mouse model, you wouldn't be doing behavioral phenotyping necessarily. You would be just more looking at the knockdown within the mice.
No, the goal there would also be to rescue behavior. I think that would be
okay.
a really, really high standard. If you could show something that's sort of Dup15q syndrome related, and you can sort of provide some therapeutic benefit there by knocking down UBE3A specifically, that would be really powerful.
And this is going to be a doozy. Do you have any idea of timing?
Timing
of kind of getting ready for an IND filing, of kind of approaching phase 1-2, trial design, kind of that stuff?
That's really hard for me to say right now. I mean,
Okay.
We're trying to push fast, so
Sure.
But timeline, that's something I can't comment on.
Okay.
Sorry, Eric. I'll make Dennis the last one.
Thank you very much. Just a stupid question. I'm not an expert with Dup15. To which degree is it understood that UBE3A is the main gene? And with your technology, are you pursuing different versions of isogenic lines where you CRISPR in and out different genes just to see if the molecular readout is really UBE3A specific? And if, just out of curiosity to understand, because you used in your talk a little bit the UBE3A the gene disorder as similar to the Dup15 disease. And I just wonder how equal this is, and if you are planning to explore that further.
Yeah, I think that's a really great question. I don't think it's a stupid question. So it's actually a very good question. That came up yesterday, I think, in terms of what's necessary and sufficient. There's certainly lots of other genes that are really interesting players or potential players. The tools for studying those, I don't even think are as good as doing UBE3A. And I think despite the roles that those other genes may play, I think it's pretty clear that UBE3A is important. So I don't, maybe that'd be a really interesting scientific question and something to follow up on, as we or after we pursue UBE3A. But certainly I think the idea is that reducing UBE3A levels should have some therapeutic effect. And we want to show that by modulating the phenotypes in cellular models where all the genes are duplicated, but also changing it in mouse where there could be some behaviorally relevant phenotype that we can.
Yeah, I mean, it looks really that your technology or your readouts at the end would enable you to look at the synaptic properties of different versions of these chromosomal region abnormalities.
Oh,
and
yeah,
I mean also from a marketing strategy at the end, you might cast a wider net if you would be able to show that, to which degree the locus versus the gene plays a role. Probably the net is wider, which might be worth pursuing, because it doesn't sound to me that it's too much of an experiment at now generating a couple lines with different chromosomal rearrangements and then just do as a proof of principle to read these molecular readouts just to see how the firing is different or same. And if it would be same, would be pretty cool, right? So you would cast a very wide net.
Yeah, I think that at some point that would be a route that's worth pursuing. I think it's not as hard as it used to be, but I don't think it's easy yet. So certainly something that's probably you're more able to tackle now, and certainly I think maybe could be of interest, but not something we're pursuing right now. Thank you.
Great, thank you.